Hi, I'm Katie.
And I'm Jane.
And today in this video we're going to be talking about the different parts of this compound light microscope.
We're going to be telling you about the parts of this microscope going from top to the bottom, and then we're also going to be showing you the parts of the DIY version that the BioBus made.
So when you get into a laboratory and you see these big, expensive microscopes, they may look really complicated.
But once we figure out the function of each part, which we're going to teach you, you're actually going to be able to design and build your very own. So let's get started.
The first part of the microscope that we're going to be looking at is called the eyepiece, which you can see right here. The eyepiece works together with the lens in your own eyes to magnify whatever you're looking at under the microscope up to 10 or 15 times.
This black tube here is connecting our eyepiece lenses with our objective lenses with something called a tube lens in between. Now the tube lens and the objective lenses are going to work together in order to create an image that a camera can pick up. In this case, our camera is here and our image will be projected onto the screen.
This particular microscope has 3 different types of objective lenses. It has a 10 times magnification, a 40 times magnification, and a 100 times magnification. You can tell the difference between all these lenses because of the size of the lens as well as all of the labeling.
When you're working with your highest power objective, which in this case is 100 times magnification, you'll see that the lens has a special property. This is called a retractable lens, and it's designed so that the slide won't shatter the glass when it's pushed up against it.
So the next part of the microscope we're going to be looking at is the stage, which is where your sample rests. The sample is held in place by this thing called the stage clip, which keeps it in place, and then to move the sample around, you don't want to use your hands, so you use these knobs on the side, so you can move around to see whatever part of the sample you'd like to. And then to move the stage up and down so you can get your image into a clearer or sharper focus, you use the fine and coarse adjustment knobs, which are on the side of the microscope right here. So the coarse adjustment knob moves the stage up at a greater speed and greater distance, and you normally use that first, and then after you get the image into a little bit of focus, you use the fine adjustment knob, because that moves it in smaller increments, and then you can bring your image into an even sharper focus.
When you're using a high power objective, the stage is going to be closer to your lens, so you're not going to want to use your coarse adjustment knob, only the fine adjustment knob. Right below your slide, you have something called your condenser lens, and this is going to collect and focus the light from your illuminator onto your slide.
And then below the condenser lens, we have something called the aperture diaphragm. This rests below the condenser lens, and this is where you can control the amount of light that illuminates your sample. It also controls your depth of focus.
And finally, we have our base, and in our base we have our illumination system, also known as our light source, which is going to shine the light up through our specimen.
Okay, so those are all the different parts of the compound light microscope, and now we're going to show you how you can take these parts and build your very own version.